Hello, everyone. Welcome to the salon. We'll have some folks joining. But in the meantime, if you'd like to join our Slido, we're going to be using Slido for this event. You can go to slido.com/DataWorks to join and participate with us today. We'll start with a poll. You should be able to see this when you go to slido.com with the hashtag DataWorks. We're just curious what types of data sharing and reuse actions you've done in your work. It's okay if the answer is none, but we want to get a little bit of audience engagement here. So go to slido.com and use the hashtag DataWorks, where you can scan on the screen to start the poll. In just a few minutes, we'll start the session. We've got some really exciting speakers today, and I'm looking forward to learning from them. Okay. All right, we've got a few folks doing the poll. Again, you can join at slido.com and use the hashtag DataWorks to join the Slido. Okay, all right, let's jump right into it. Morning, everybody, or good afternoon. My name is Emily. I'm the program manager for the DataWorks program, and I'm going to run through a little bit of who we are, and then we'll jump into these speakers. So FASEB is a federation of scientific societies representing about 110,000 scientists across many disciplines. Our members you can see here. We are here to support our members and the biosciences community to learn about data sharing and reuse. So the two goals of the DataWorks program are to link researchers to expertise around data sharing and reuse. Talks like these are a way that we do that to bring people who can share what they've done in this field so that you can learn and implement that in your world. And also to bring together people and create a space to improve the ecosystem, because we know that the biosciences is really moving quickly and in some exciting ways towards data sharing and reuse, but there's a lot to learn. So we want to be a convener for that. We have 3 programmatic elements. We have a prize. You'll hear from one of our prize winners, Arjun Krishnan. We have the salon where you are today, and then we also have a help desk, which provides resources for you to learn about data sharing and reuse and understand how you can do that in your own science. So with that, let's get into today's topic. We've got metadata in biomedical research, and we'll begin with one quick poll, and then we'll jump into the conversation. So what words come to mind when you think about metadata? This is a broad question, but you can join on slido.com using the hashtag DataWorks and see what comes to mind when we think about metadata and its role in scientific research. Curious if we have anybody who's gonna jump in. I know it's a broad question. Understandable. All right, I like that. Any more thoughts about what comes to mind with metadata? Transparency. Yeah, data about data. Searchable, fair. Yes, these are all great ones. We're gonna have a really good time today. Appropriate reuse, yes. Yes, music to my ears, this is. I love this. Great. All right. Controlled, interoperable. DataWorks. All right, here we go. All right, well, now that we've got our minds on metadata, I'm going to stop sharing and hand it over to our first speaker, who will be Arjun Krishnan from the University of Colorado Entrance Medical Campus.
I'm assuming you folks can see my screen. All right, great. Hey, everyone. Uh, Good to be here. Thanks for the opportunity, Emily. And what I'm going to talk about today is a bunch of work that we've been doing over the past few years, thinking a lot about removing metadata barriers to make publicly available omics data as easily discoverable and reusable as possible. Okay, so this is what we want to do. And I'll begin with some motivation. Um, First, I want to give a huge shout out to the wonderful group of people that get to work with every single day in my group. These are graduate students, postdocs, programmers, undergrads, high school students. They're fantastic, and these are the reason I get up in the morning and have to work. We generally are a very freewheeling lab. We develop lots of algorithms to answer many cool questions that are related to biomedical data science, and we are roughly always thinking about how can we gain more and more and better in our understanding of genetics and molecular mechanisms or cellular mechanisms of complex traits and diseases. We want to develop methods that are general purpose, that are agnostic to the trait or disease that we're studying, but generally, keep developing, do this in some data-driven way. And what I mean by data-driven way is that our lab's focus is to take advantage of publicly available data to do this. Okay, so I'm not talking about data sets that we get exclusive access to because we are collaborating with some specific researcher. Okay. So this is data that all of us can actually, in theory, get access to and work with. So what I mean by this is, massive amounts of diverse public data collections. This can be public or say a bulk or single cell omics data that have been generated in the past 25 years. And there are millions of samples. These could be molecular interaction networks that people have mapped out through high throughput experiments. And there is also lots of unstructured descriptive information. Some of this is metadata, and some of this is literature, which is people describing the scientific knowledge in the form of papers. And then there is also plenty of population or individual level genotype phenotype data. So we work with all these types of data sets that are public data sets. Okay. And then we always want to do what we want to do is to anchor them on high curated, high quality knowledge bases or ontologies. This is experts' current abstraction of our knowledge in these fields. Okay. And what we do in our group is to say, okay, how can we take advantage of these different pieces of data in different combinations? And what we folks in my group develop novel algorithms, benchmarks, and software to use these data to advance every step of data-driven discovery. What this means is that we develop methods to harmonize data. For example, data could be generated from completely different platforms and technologies. We want to see how do you reconcile these data sets so that they can become comparable to answer your questions. We then develop methods to say how can you take this data and create very comprehensive data representations of the behaviors of the entities that we are measuring. It can be genes, proteins, or metabolites. Then we develop, machine learning models that can take advantage of these representations and then make predictions about individual genes or combinations of genes in the context of, traits, diseases, functions. We also think a lot about benchmarking, which means that even though we are developing methods, we want to make sure that the community also gets to develop methods and they are able to compare methods to each other in a fair way. So, which means that we release benchmarks so that others can use the same data sets to compare, different methods in a uniform way. And then we also write a lot of software and then release web servers also. These web servers are especially not databases, which means that they're not retrieving data, but they are interfaces for biomedical researchers to run custom machine learning algorithms. So, which means that they provide an input. We spin up a cloud instance where we train a new machine learning model based on massive data, and then get new predictions for this particular user and get them back to them. Okay. All right. So to give you a sense of the scope of this data, which I think all of you are very familiar with, let me just talk about omics data, right? So if I went to ArrayExpress 3 years back, okay, these numbers are already dated. And when I was looking at human data, if you look at human data, if you take the 14 most common types of omics experiments, it clocks to about 26,700 data sets and more than 1.3 million genomic profiles. And this is just within this particular resource. There are several other data. This data has been generated by researchers in the past 25 years and deposited to public databases, okay, like NCBI, GEO, ArrayExpress, SRA, and so on. And these data are incredible because these are samples that come from hundreds of tissues and have been studied in a variety of contexts, including genetic, environmental, physiological, and clinical factors. So I think all of us are on the same page here. I'm literally preaching to the choir saying that wouldn't it be awesome to use all this data, right? And now this is just human data, right? Now, if you expand this to data from these major model organisms like mouse, rat, fly, worm, and fish, this number is close to 2 million profiles. In fact, this number is way larger than this, but I won't go into these nuances. Okay. Now, the problem that I want to talk about today, again, this problem, I think all of you are super familiar with this, is that biomedical big data has a serious metadata problem. And this causes these public data, although they are incredibly valuable, they are acutely underused. And this problem comes in two different flavors. The first is that the metadata, which is data, somebody said data of data, right? So it is highly unstructured, which means that there are descriptions of samples and experiments that contain information about the source and in form of some ambiguous plain text. Okay, I'll tell you what that is in a second. The second problem is missing metadata, which means that although somebody has written a description about a sample or a dataset, it doesn't mean that they have included every attribute of the sample or dataset in that description. So which means that sample descriptions are frequently going to miss key basic pieces of information about the source, okay, or the experiment. Now because we are so interested in using data effectively, we started also, developing methods to tackle these two problems. Right. Okay. First, I'm going to talk about some of the work that we've done with unstructured metadata. So just to get all of you on the board, this is what, this is a typical description of a sample. It's just blocks of text. Sometimes it is many phrases, that are attached to some keys, but you should just think of this as, unstructured text and, bags of words or bags of phrases, literally. Okay. So that's the first challenge. The second challenge is that, all of us biomedical researchers and research any disciplinary experts, we use very different terms to refer to very similar things or even the same things. Somebody might say enteric, others might say intestinal, they mean the same thing. Somebody say myocardial infarction, somebody else says heart attack, they mean the same thing. There are also going to be sentences like this. So let's say you're looking for which tissue was the sample generated from. You're going to see a description like this. As humans, we can actually read this and exactly tell that macrophages was the source of the sample from which omics data was generated. But if you do plain text search of names of tissues and cell types, you might hit many different things. Okay. There are also cases where somebody will say they study psoriasis, but they will never mention the term skin because that's the sample, that's the source from which the sample was obtained. Same with cerebral palsy or muscle. And then, of course, there are going to be so many typos. Okay. So we have to deal with all these problems. Right. So this work was spearheaded by Nat Hawkins, an amazing graduate student who finished and is working as a data scientist in the industry, and then Mark Moldauer, who was an undergrad who started this work, and he's now a medical student. And these two essentially came up. We figured out how to deal with this by taking advantage of really cool developments in computer science and natural language processing. And I'm just going to tell you the basic idea. If you want more details, I'm happy to chat during Q&A. The idea is the fundamental idea that we're using is called word embedding. Okay, the word embedding, the idea is very simple. The idea is to say words are very hard to provide as input to statistical and machine learning algorithms. Therefore, we're going to turn words into numbers, and the numbers are easy to provide as input. And the way these numbers are generated is by training a large neural network model that can consume a massive amount of text. Okay, just lots of text. For example, all the text in the internet or all biomedical papers, and then figure out a way to represent every word or every phrase as a set of numbers so that if two words mean similar things, you can more or less be guaranteed that they will have very similar numerical representations. And the vice versa is also true. What we used in the publication that we had 1 year ago. What we used was a different model. It's called FLAIR. It does something called character embedding, not exactly word embedding. This has a few advantages, And we are also trying out other transformer models. I'll talk about this a bit later. Okay, now the approach that we did is the following. We actually went ahead and manually curated nearly 12,000 samples that we obtained from GEO to various tissues in the body from which the samples were obtained from by reading the description. Okay. And now we have essentially labeled samples and their unstructured descriptions. So that's what I'm showing you on the left. With this, what we did was to take the description of every single sample, do some pre-processing, and do something called sample embedding. Okay. So what we do is to take the text description of the sample and then now create an embedding where every single sample is now represented by a bunch of numbers. These numbers were obtained by taking the embeddings of every single word in a sample's description and then combining them in some intelligent way. Okay. We are not doing exactly averaging, but you can think of it as averaging for now. So now we have a numerical representation of every sample, but based on the description of the sample. Right? I'm hoping that all of you are with me at this point because I'm just seeing black boxes, so I'm not really sure what's happening. Okay, all right. Okay, now once you have these numerical representations, what we then do is to train a machine learning model that can predict whether any sample based on its text-based numerical representation, whether it comes from a particular tissue or not. So you can train a model of a heart, a brain, kidney, liver, and as many tissues and cell types as possible. Okay. And once you have done that, then you can take the unstructured description of any sample, do the same pre-processing, turn them into numbers based on the description, and then provide them as input to the pre-trained models and get predictions about whether a given new sample based on its description alone, whether you can predict whether it came from a particular tissue or not, okay, and for each tissue or cell type of the body. So this is the work that we published 1 year back. And this works quite well, okay? Here I'm showing you another approach for doing metadata annotation. Okay? This is to take, again, unstructured text and associate them with specific annotations, maybe tissues or diseases or whatever. This is called meta-SRA, and it's actually a really cool method. It's based on graph-based reasoning, and the approach is completely different from the approach that we're taking. Okay? It uses ontology structure and knowledge consistency to create these associations. I'm showing you performance in terms of average precision, and you should, if meta-SRA has a median performance of 0.5, it means that if meta-SRA says here are 100 samples related to the liver, 50 of them are likely to be correct. That's what it means. Okay. And then the performance is pretty broad. There are some tissues for which meta-SRA does really, really well, and there are other tissues for which it does pretty poorly, but on average, it's okay. Those are the points that compose this box plot. Every point is a tissue. Now, here's the performance of our method. We naively called it NLP-ML because it combines natural language processing and machine learning, and we get 75% right most of the time, right? And the range of performance is actually pretty much narrower, so which is a good thing. Right? Now, I want to tell you that if you see performance plots like this in papers or talks, you should always say, okay, doesn't mean that this method is always better. There are some cases in which most cases this method is better, but not all the time. So for example, here's a scatter plot where every point is a tissue. The previous method is on the x-axis, our method is on the y-axis. Most of the time it's equal or better, right? Our method is better. But I'm showing you only a subset of tissues. Here are all the tissues for which we did this analysis. On the leftmost are tissues for which we had very little training data. So in this case, there are going to be many tissues for which the previous method actually does very well, and we perform very poorly, right? And then, of course, there are many tissues that are the far right, where there is enough training data that we perform consistently better than the previous method. So it's we need to keep this in mind when we interpret results like this. All right. So, of course, when there are two methods that take very different approaches to solve the same problem, you can combine them together. And the combination of our method with meta-SRA gives us very good results across the board. Okay. All right. And the accuracy, actually, the average precision is very high. So which means that most of what these two methods together tell you are going to be right in terms of metadata annotation. All right. We are also always very interested in interpreting machine learning models to make sure that they are picking up biologically meaningful signals. So what we did was to take the machine learning model that was trained for every tissue in the body and compare them to each other. And our expectation is that if two tissues are very related in the body, they should also, the machine learning models corresponding to those tissues should have used very similar signals in the underlying text to make those predictions. So that's what we did here. Every point here is one tissue machine learning model, and then they are close to each other if their models are very similar to each other. Okay? And then we see that there's rough clustering based on organ systems of the body, and that's how we know these signals the models capture are meaningful. All right. The final point is that here's the cool part, right? Sample description is sample description, irrespective of the omics technology. So we created our gold standard by reading the descriptions of microarray samples and annotating them to various tissues of the body. But once the models are trained, they can make predictions on descriptions of any omics samples. It can be proteomics, RNA, ChIP-seq, methylation, whatever it is. So we have essentially been taking these text-based models and applying them to descriptions of many different omics data types, and we're getting pretty good results across the board. All right. Like I said early on, we are also passionate about software, which means that we want to make sure of two things. One is that when we produce a publication, any other researcher should be able to replicate everything that we've reported in a paper, which means even reproducing the figures, okay, right from the beginning. And they should be able to use the software to build on top of it, which means that they can modify the software, add extensions, and develop new methods and so on. So to satisfy both, we released Text2Onto. So Text2Onto is a GitHub repository, which is very detailed. It has excellent documentation, and it has two very nice utilities that you can use to build on top of. All right. We're doing a bunch of other things right now, which means that we are actually taking advantage of really new transformer-based language models, getting larger training sets, and really focusing on interpretability and explainability. Somebody in the Slido cloud basically said, interpretability are all the things that are important, and then we also take this seriously. I talked about tissue classification of sample descriptions. We're also, of course, doing this for phenotype diseases and all that stuff. We're expanding this to more species, so not just humans, but other model organisms. And we're building a web server right now where we're going to put all the predicted metadata, structured, standardized metadata that we predict for all samples that have ever been generated and deposited in public databases, and put those predictions in a single web server so that somebody can actually search and find relevant datasets and samples as fast as possible based on some standardized annotations. All right. I'll give you a hint of one, improvement that we're making, which is that in this, I showed you this whole workflow, and then in this workflow, one of the challenges is that we take the whole description and turn them into a set of numbers and provide them as input to machine learning models, right? I'm sure it hit some of you saying that, okay, does it really make sense to mush the entire description into a single numerical vector? It mushes up different types of terms that could help in doing different things, telling you different things. And of course it also contains many uninformative potential words and phrases that you're again combining and maybe even reducing the signal of the embedding a lot. So of course we are cognizant of this. So what we have been doing now is to actually improve on that front. I'm not going to exactly tell you the details of the method in the interest of time, but here is the performance of the published method. And now we are developing methods that actually preserve words and phrases within the description in terms of either words or word clusters. And then I'm going to show you the performance. Okay, so based on words and word clusters, we're already getting much better performance than what is already published. The same with phrase or phrase clusters. This is for tissue classification. Again, for disease classification based on phrases or words, we get equal or better performance. The huge advantage is that now the models are even more interpretable. They are both interpretable and explainable. That's the most important thing. They are interpretable in the sense that we can go to the machine learning model and understand the features way better. And second, when we make a prediction for a single sample, we can exactly tell you which pieces of words or phrases were used to make that prediction, so that you as the user can look at this and really say whether the model is performing well or not, right? Or whether to trust the prediction or not. All right. Okay, maybe this is worth showing you. So I created this table to show you how awesome these models are in terms of even picking up words that are biologically or biomedically related to what you're searching for automatically. So for example, models, when we train a model to predict whether the sample corresponds to trachoma or not, it automatically recognizes, squamous metaplasia, matrix metalloproteinases, immunofibrogenic disease process as key, pieces in the text that are going to be informative for trachoma. Okay, the same thing for many other things. So on the right in the blue are the features in the text the model automatically picks to make the prediction on the left. And you can see the text is totally different on the right and left, but still it understands relationships. automatically. All right. Okay. So, Emily, can you tell me how much time I have beyond this? Based on this, I want to.
Probably about 3 to 4 more minutes.
Okay. All right. So let me go through this second part, very quickly to give you a good idea. So up to now, I've told you how we deal with unstructured metadata, right? So, which is using an NLP-based method to extract standardized annotation, or rather infer standardized annotations. The second part is missing metadata. If the information is completely missing in the text, you can't do anything about it. You can't apply NLP, right? So what do you do in that case? We encountered this problem because one of the biomedical questions that we are very keen on understanding is this idea of sex and age specificity. And then what we thought was that we'll go to public data and look at sex and age specific data, and we realized very soon that there is a widespread lack of basic information in all these things. I'm sure Mark is going to tell you about how age, even if it is there, it's encoded in so many different ways. Okay. So even if you account for the fact that they are encoded in so many different ways, they are encoded at all only in about 20 to 30% of the samples, which is crazy. Okay. Same with sex. And what I'm showing you as bar plots are data from various technologies. Right. So this is pretty bad. So what Kayla Johnson, an amazing graduate student in my group, decided to do was to actually we focused on transcriptomics data. And from transcriptomics data, we also have the expression profile of almost all the genes of the genome, right? So what she decided was to predict missing metadata from the expression profile. Okay. So do this, she actually did a massive curation of data. I'm not gonna tell you the details. Ignore the top part of the slide. The bottom part is the most important. She essentially curated 600 studies that clocks about 30,000 samples, equally spread across microarray and RNA-seq, and equally spread across females and males. And she's annotated them to females and males. And second, these samples, these 30,000 samples, also come from almost every age group along the human lifespan in both females and males. So we have very good data representation, and this was a massive effort on her part. And now what she did was to essentially do a couple of things. I'm going to skip over some of these results. It's where what she's been able to do is to take the data and look at sex-specific genes and then use them to do sex annotation, which is very straightforward. Everyone knows this very well. It's not novel. Okay, but what we are able to do is to do sex-specific sex annotation in an age-specific way. Okay, this is what is novel. So nobody has done this in an age group-specific way. Let me. Okay, so here is the most important part about the metadata. Age group prediction has always been very hard, but then Kayla has accomplished this pretty well. So what we do is to take the human lifespan, divide this into 11 age groups, and then what she does is, for example, I'll take fetus as an example. She would treat all these samples as positive examples, everything else as negative examples, and then train a machine learning model that essentially takes the gene expression profile of every sample as input, and then predicts whether the sample is the fetal age group or not. And then once you train this on a massive number of samples, you are essentially able to get a model that does, prediction. I'm going to skip over the prediction performance. Let me just tell you one thing. Okay. All right. So our prediction performance is actually pretty good considering how hard the problem is. And this is especially good for this one result that I want to show you. Remember, I told you that the way Kayla sets up this model is she says that I want to predict one age group versus the other age group. She never encoded the fact that these age groups are chronologically related to each other. And yet, the model correctly captures this chronological relationship, which means that it provides a model that is trained to predict fetus samples, will give the highest probability of fetus samples, but then the next highest probabilities to samples that come from infants and children, and so on and so forth. It somehow captures these chronological relationships, and that's how we know these signals are biologically meaningful. And we do this across the board. Okay? So let me skip over this part and show you just one final thing. Okay. So we have a bunch of different projects that predict missing metadata based on the expression data. Okay? And together, we have two things, right? One is text-based models that add standardized annotations. Other is expression-based models that add standardized annotations. For example, here I'm showing you the performance of the expression-based model. And these two are totally complementary. One is using the text description, other is using the omics molecular data. So therefore, if you actually combine them together, you get even better prediction. And these are, super, very good annotations that we get. All right. So that's it. So I've told you about these two problems and how we are addressing it. And they are described in a publication and a preprint. One is based on NLP and ML to work with text. Another is transpronoun-based ML to work with missing metadata. All right. You want to stop here and then take questions?
Yeah, thank you, Arjun. That's very exciting examples of how we can move through some barriers to metadata and find some really great findings. We'll have questions. You can post them on the Slido, and then we'll have some time at the end. But I want to hand it over to Mark Musen, who is based out of Stanford University, and we'll be talking about metadata, making biomedical data fair. Thank you, Mark. We're seeing you outside of present mode.
There we go. I'm sorry. I didn't realize we're okay. Sorry, bad day. Hopefully, we'll have a good opportunity now to talk about really a complementary problem to what Arjun talked about. What I want to discuss is really not necessarily how to deal with all of the horrible metadata that are already out there, though I'll mention that. But I think what I'd really like to talk about is, as investigators, how can we try to make our metadata better at the source so that we can begin an enterprise where science puts data sets online in a way where they are actually useful for all the things that people talked about. This group obviously understands the FAIR principles, and you identified that as really an important element of why we want to put metadata online in a good way. And although all of us talk about the FAIR principles, none of us really look at them very carefully. It's pretty overwhelming, actually, for most investigators. But when you look carefully, they're all about metadata. And what makes data findable, accessible, interoperable, and reusable is really the fact that the metadata are decent, that they are searchable, that they are useful for understanding what the investigator has done. And the challenge is that, I mean, as we know, is that most of us view our work as publishing papers, not leaving a legacy of reusable data. And no one really wants to pay for data sharing. And creating good metadata is terrible. It's hard. And that's why when you look online and you look at what kinds of data sets we have in repositories such as Biosample, you see stuff like this, which is absolutely useless if anybody wants to make sense of what someone has done in an experiment, and is absolutely useless if we want to be able to make new discovery from other people's work. And so what do we need to do? Well, we need to be cognizant of the fact that one of the first things we need to do is to use standardized terms so that we can do retrieval of data and integration of data in a coherent way. Arjun told you that I'd be showing you this slide. It's one of my favorites. It shows you how in Geo, there are like gazillions of ways to describe the age of a subject, and not even the age of the subject, including all the possible typos. And so this has to be represented in some sort of standardized way so someone can do a query. The other side of the coin is that not only do we need standardized terms, but we need a standardized structure. And probably the best example of that really is what happened in the microarray community 20 years ago, when it was recognized that there needed to be a standardized way of creating what's called a reporting guideline for microarray experiments. People had to know what was the substrate, what was the platform, what were the experimental conditions. And that led to Miami, which provided a standardized way for talking about what are the aspects of the experiment that need to be included in any kind of a coherent description of a microarray experiment. And as many of you know, it didn't stop with Miami. We have all kinds of standards in biology, like MIATA and Miriam and MiniMess and Miss Fishy, and really a couple hundred of these. And is exactly this kind of community standard for describing the attributes of an experiment that needs to be standardized in the context of a reporting guideline, so that we know how to query data sets and we can get the information that we need in, so that we can integrate my T cell assay with your T cell assay, or otherwise we're going to be stuck. And so if we really want to have data sharing, if we want to have fair data, we need ontologies that provide the controlled terms. We need reporting guidelines like Miami that provide a standardized structure. We need technology to make it easier to author good metadata in the first place. And I'm going to gloss over this in this talk, but it's really an important component. We need community standards, and we need procedures to create those community standards so we have structured ways of talking about the kinds of experiments that we do in biology. So our approach at Stanford has been to work on technology to address these problems. One of our initial forays in this area has been the creation of technology called CEDAR, which encodes community-endorsed reporting guidelines as templates that give you a fill-in-the-blank authoring opportunity, so to speak, to create structured metadata that adhere to that reporting guideline without even having to know what's in it just when you're starting out. And we obviously use ontologies whenever possible to provide standardized values for the template fields. So let me just give you a flavor for what this looks like. You go into CEDAR, and you can create a new reporting guideline in the form of a template, or you can see what templates you already have in the library. Suppose we're interested in a template that gives us the ability to use the data dictionary and structure expected by biosample. We're talking about human samples. We select biosample human. We say we want to populate the template. We can see what the template looks like. And what we see is that the template captures what goes into biosample as attribute value pairs, where the names of the attributes are the reporting guideline, if you will, for data that have to be put into biosample. And CEDAR gives you the ability to fill in that template. We say the sample name is 56, the organism is Homo sapiens, the tissue is whatever we were about to specify. And note, we don't just type in any old tissue. We get a drop-down list. And in this case, it's all the kinds of tissues that are enumerated in the Uberon ontology for anatomy. And it's because the creator of the template has said that Uberon is going to provide the best resource imaginable for enumerating tissue types. And this is the one that the template designer says this is the one that we should use in our experimental community for describing tissues in a standardized way. And so and also note that the drop-down list is not, well, it is all of the tissues that are in Uberon, but they're sorted in the order in which those tissues have been specified in previous metadata for biosample when the organism has been Homo sapiens. And so because we have a database that caches all the previously entered metadata that have been put into CEDAR, we can use that to identify patterns in metadata and use that to simplify the authoring of new metadata, trying to make the burden of metadata authoring much less. Note also that because we can learn from these data, we can say if the tissue is lung, then these are the possible diseases. And they're sorted so that when the tissue is lung cancer and COPD and squamous cell carcinoma of the lung are the ones at the top of the list. When we say the tissue is brain, then we get the list sorted so that brain diseases are at the top. And again, it makes the authoring of this simpler. So in its usual form, CEDAR uses these web interfaces to allow investigators to create structured metadata that adhere to community standards for reporting guidelines and ontologies just by filling out these forms. And we've used CEDAR in a wide number of NIH-sponsored initiatives. We've also are working now with the Open Science Framework and with Dryad to start moving CEDAR into generalist data repositories, recognizing that's probably the most important customer that we might have for this kind of technology. And we're really excited about the ability to take these technologies and make it possible to create reporting guidelines and ontologies in very easy to use forms that can be made available to investigators at the time they're creating their data sets. One of the organizations with whom we're doing a lot of work is HubMap, which is an NIH-sponsored common fund consortium that is trying to identify biomarkers in basically every cell type in the body. And we've created really nice templates that allow people in the HubMap initiative to be able to fill in these templates and create in a structured, standardized way everything they might want to say about, for example, the samples from which they're collecting cells, the assays that they're performing, and so on. But I just want to tell you, the HubMap people looked at us and says, we don't like web forms. We're not going to use web forms. We like spreadsheets. And like many investigators, they said, don't give us a mechanism that requires us to fill out spreadsheets. Give us something that, fill out web forms, give us a spreadsheet. And so we've actually worked out a new system where the CEDAR templates that one authors in the web interface that I showed you earlier can now be translated into an Excel spreadsheet. And the Excel spreadsheet actually embodies all of the value sets that are derived from the appropriate ontologies that are needed to ensure that the value types are correct. And what we create is a spreadsheet that basically allows one to enter metadata that comport with the predefined reporting guideline using the appropriate ontologies. And this is what the spreadsheet looks like. The columns in the spreadsheet are all derived from the metadata template entered to CEDAR. And this is really great for people who have multi-valued, multi-data from multiple experiments. The problem with a spreadsheet is, of course, that although in our case, we can try to provide mechanisms to encourage users to have the correct data types for their metadata values, we can use ontologies to create the correct terms. A spreadsheet is open-ended, and Excel doesn't really enforce that that's done correctly. So we've created this metadata validator where you take your spreadsheet that has been created using the CEDAR template, you drag it into the metadata validator box, and the metadata validator allows you to identify where there are errors, and it actually will suggest repairs for those errors because it understands what the template looks like, and it knows what the metadata should be, and it suggests those changes. The user can very quickly repair any problems with the spreadsheet and then upload that spreadsheet to whatever place that metadata has to be stored. So basically what I've told you really quickly is that when we think about scientific metadata, there are two kinds of standards that really matter. The ontologies that allow us to control the terms that are used in metadata descriptions, and reporting guidelines that enumerate the aspects of the experiment that need to be specified for somebody to make sense of what has been said in that, what been done in that experiment. And really, if we care about fair data, if we care about being able to reuse data, we need to be able to have reporting guidelines and ontologies that are relevant for all the areas of investigation that we're concerned about. We need to render those metadata, I believe, as templates that can capture the reporting guidelines. We have to have control terms in appropriate ontologies. And we need technology like CEDAR and like this spreadsheet system that will allow investigators to make sure that their metadata at the start are fair and that their data sets are going to be reusable and valuable to the entire scientific community going forward. And let me stop there.
Great. Thank you, Mark. We can have questions either come through the Slido or you can raise your hand and ask questions. I think we've covered the real breadth of the spectrum of metadata, and I'm excited to hear Ian's questions. I do see one within the Slido around how to find standards for an experiment. Is that something that comes from the journal or repository? It seems like they're not clear. And then we have a second one around, is CEDAR freely available?
Yeah, let me answer that. CEDAR is freely available. It's open source. I think it'll probably have its greatest impact when it gets fully integrated with the generalist repositories, but it's available now and lots of people are using it standalone. The real challenge, as you're implying, is knowing what standards to use. And actually, the bigger problem is actually not a surfeit of standards and overlapping standards, but frankly, we just need a lot more. I think we really need to encourage people to do what the microarray community did and recognize that they need a community-based standard in order to be able to make sense of one another's work. And the good news is when you go to the CEDAR template library, you can see the existing standards that people have entered, and you can get descriptions to get a sense for which one you should select. But I really think the challenge for the scientific community is going to be more identifying where there are areas of investigation that have not had any thought about what really should be reported, and really getting people engaged in creating those standards in the first place. I was really encouraged by all the positive comments that you all had about metadata at the beginning of the hour. Most of the time when people talk about metadata, they use words like boring or valueless. And I think the fact that this community understands the importance of metadata is really very gratifying.
Yeah. We have a comment here from Sun Young Kim who says encouraging data repositories to adopt metadata templates is something that is exciting. Can you speak to the ways that you see that moving now and where you hope it goes in the future? I know you talked about integrating CEDAR into some of the generalist repositories. But
yeah, well, I certainly see that happening to the greatest extent in communities that recognize that their data need to be integrated and shared. So an example is the HubMap community, which is, as I said, an NIH consortium that's doing work to identify biomarkers in every human cell type. They didn't need any encouragement. They knew that they had dozens of laboratories around the world all working on this problem, each of which were contributing data sets to a central repository, and that it would be impossible for anybody to make sense of the data unless there were standards. And so very early on in the project, they created a data coordinating working group. They worked on metadata standards, and then came to me afterwards and said, we need some technology to make this all more feasible. So I think it's going to be a matter of scientists recognizing the value of data sharing up front that will do it. The other thing that will encourage data sharing, frankly, is that we know that OSTP has told all federal sponsors in the US data sharing is important and data have to be online. We know the European community has become very, very active in requiring open science and data sharing. So in some sense, the funders are going to say thou shalt, and that's coming. And when that happens, there'll be a lot of incentive, of course, to make data available. And it doesn't make any sense to make data available unless we make them available in a useful way.
I have a second question here about integrating CEDAR with fairsharing.org.
That's a great question because fairsharing.org stores guidelines, but it stores them in a textual format. And we'd love to have it, it would be a great project to actually make more of an interaction so that the computer processable guidelines that we use in CEDAR could be available in fair sharing. That we just haven't gotten around to doing that. And that'll be, I need to talk to the Oxford folks about that possibly.
I'm curious if you could talk to, I know you're not from Fair Sharing, but if you could talk to just in a general level what that resource is and what it provides to.
Yeah, thanks, Emily, because not everybody knows. So Fair Sharing is a resource that was developed by the eScience organization at Oxford that is a repository that points to existing data repositories, that points to reporting guidelines, really for all science, but biology dominates because that's frankly where most of the reporting guidelines are. And it also points out ontologies that might be useful for creating structured metadata. But all of this is in text. And so what fair sharing doesn't do is provide a mechanism for a tool like CEDAR to be able to get access to a machine readable version of the guideline and apply it. That's why we have to have a separate library in the CEDAR work. Yeah, okay. Good question. So the clinical data, the common data element repository at NIH is really important. We have actually done work with NCI because they have their own CADSR repository for common data elements. Common data elements being really ways for talking about data in a standardized way. So in some sense, a CDE is a piece of metadata that describes what should be said about an element of data with a standard way for asking questions that elicit the data value, ways of identifying what are the value sets that are relevant for that CDE. And yeah, we've actually, all of the NCI CDEs are integrated into CEDAR right now. And actually, we're actually working on getting all of the NIH CDEs into CEDAR because we think that's really important.
I'm curious, I would like to hear from both of you, if there's other questions from the audience, but just more broadly, where you see data sharing being enabled by metadata and where you see that moving in the next year or two, that you see that there's a lot of energy and the community is moving in that direction. Just curious.
I'll give Arjun a chance to answer that one first.
Thanks, Mark. And my answer will be not surprising at all, given what I was talking about. So I think clearly language models have become so advanced, right? So I think one of the areas in which metadata annotation is going to become very good, especially both in annotating pre-existing data and also annotating new data, like Mark is talking about, is, for example, I'm envisioning a system where it's not somebody going to a spreadsheet and then selecting terms from a drop down and then populating this, maybe just uploading their abstract and paper, and then we automatically identify structured terms and then provide them for confirmation to the user rather than having them enter and select. So some combination of the expert who wants to curate it working with a large language model using like some using some chart interface is probably the meeting point. So I very safely, decided on something that's an intersection between my work and Mark's work.
Mark, what do you see coming forward?
Yeah, I would agree with that. I think it's going to be twofold. One, there's an enormous interest in trying to clean up existing metadata, and the kind of work that Arjun described here is really important because, frankly, there's just so many terabytes of existing bad metadata that they're crying out for someone to try to fix all that. The biggest challenge, I think, is going to be trying to create cultural change in science. I think all of us give lip service to fair data. I think all of us feel really complacent and happy when we put our data online. But it turns out when those data are not useful to anybody, they're not exactly going to be helpful. They're not going to be able to promote the kinds of things we're talking about in these meetings. I think the culture is going to change. It has to change because we're having too much success for people who are able to do secondary analyses of online data sets and show interesting things and important things. And the funders are demanding it, and the government's demanding it, and professional societies are demanding it. And eventually, I think the same way that we now recognize the importance of peer-reviewed publication, which 200 years ago didn't exist. I think we're now going to see a shift in the community, and we're going to recognize that publication of data is important. And in fact, data is the real output of investigation, not PDF files.
I love that. Yeah. We have a question here. Can you talk more about metadata validators? And is that something that CDER is creating for biomedical data?
Well, probably the best, yes. I mean, in some sense, the spreadsheet work that I showed you is a kind of metadata validator that's validating proposed metadata at the time of upload. And my group is actually also looking at the problem of trying to validate and clean up existing metadata using a lot of the techniques that Arjun talked about. I don't think we've gotten as far as he has, but we think it's really important for work in that area to proceed because, as I said, there's a hell of a lot more metadata out there that is terrible that's already online than is going to be created in the next year.
We have a question about using, I'm going to say OMOP, but it may be OMOP, common models, and why CDERs over them.
Okay, I view OMOP, and maybe I need some help from the questioner. I view OMOP as a way of structuring EHR data so that it's available for reuse. But I don't think OMOP has the metadata element that allows people to know what's in the data set. We don't know, just by having an OMOP file, we don't know, does this file include patients who are readmitted to the hospital early? Does this data set include patients who have had complications of surgery? Those kinds of meta statements about the data don't really fit in a standardized way into OMOP, to my knowledge. And so I think OMOP is really important as a data standard, but I don't think it helps standardize metadata. And CDER is working on trying to create data descriptions that are in a standard format rather than standardizing the data themselves.
Okay, we have time for one more question if there's any more in the audience. Then I will wrap things up here with just 2 DataWorks opportunities. Okay, behavioral studies. in lab animals, metadata standards for behavioral studies in lab animals.
I'll just say that I was recently on a panel that was convened by the National Academies looking at standards for the behavioral sciences, mainly looking at ontologies and recognizing the general problem. I think there's a real opportunity here because one of the conclusions of that panel is that the behavioral sciences is an area in particular where there's an enormous amount of overlap in the way people describe existing phenomena. And I would really recommend that for people interested in behavioral sciences, a report that came out of the academies about, well, in the end of last year, which really talks about those details in a way which I think is really, really helpful.
I know we're coming right around time. We have a next question about NCI symposium and cleaning bad data being called a graveyard. Working with I4 or Stratus to clean up that bad data. Art, do you have a?
I'm not involved in any of those collaborations. I'll defer to Arjun.
I don't, but these are great ways for us to explore this. We will take a look. Thank you.
It's definitely a graveyard.
Yes, that much is true. Yes.
Okay. All right. Well, just looking at time here, I do want to mention two opportunities that we have within the DataWorks world. The first is that we have a call for beta testers for our help desk resource. And through that resource, we are looking for people to help us understand, to run through our help desk and provide feedback and beta testing around how it can be used to support you if you have or are going to create an NIH data management and sharing plan. You can email us if you'd like to participate as a beta tester. I know it's a broad ask, but a cool opportunity to try a tool as we're putting out new features to support you. And then lastly, we have our next DataWorks Salon on November 15th. We'll be talking about something very parallel to this, which is how we can accelerate scientific discovery through data integration tools and software. So I'm looking forward to that conversation, and you can sign up and register at FASEB.org/DataWorks. I want to thank everybody for their time, and